Hello, everyone. Welcome to our last episode of this podcast initiative. This is the fifth episode, and I've called it Digging into Deep Time, How Paleontologists Study the Past. And we're joined today by Dr. Rod Taylor. I'm going to let him introduce himself soon. I'm just going to introduce what the episode is going to be about. So we're going to talk about traditional paleontology and casting, as well as modern research methods like computational fluid dynamics, 3D scanning, mathematical models, etc. Basically, how scientists uncovered the secrets of life from half a billion years ago. So this episode takes you behind the scenes of paleontology, revealing the tools and techniques researchers use to study fossils at Discovery Geopark. So in this case, our group. From traditional fieldwork, as I said, to high-tech scanning and computational, more like fancy methods, we explore how modern science is helping us piece together the story of Earth's earliest animals. So how are you, Rod? I'm going to let you introduce yourself.
Fabulous, thank you. My name is Rod Taylor. I'm a paleontologist working with the MUNPaleobiology research group at Memorial University in Newfoundland. I'm also a science interpreter at the Johnson Geocenter, so I have an opportunity to teach about the science that we're doing, as well as actually working in the field and doing real contributions to science, which is a nice combination. That's me.
Okay, well, thank you for joining us, Rod. And yeah, if you're okay with that, we can just give it away and start with the first question.
Okay.
So the first question is, paleontology has traditionally been about digging and studying fossils in the field. That's kind of like the preconceived idea that we have of a paleontologist, right? Someone wearing a hat under the burning sun and just digging. Can you describe what fieldwork looks like at Discovery Geopark and how fossils are documented and collected?
Well, fieldwork at the Geopark very rarely involves a burning sun.
Yes, very true.
Right away. That doesn't happen very often. Working on the Ediacaran paleontology here in Newfoundland it's probably quite different than what most people would envision when they think about paleontologists working in the field. Traditionally, paleontologists, they find bones, or they find shells, or they find body remains, they find tree trunks, etc. And they spend a lot of time actually extracting this material. But because this material is so rare, the Ediacaran fossils here in Newfoundland and at Discovery Geopark in particular are so rare, it's actually illegal to collect them from the field. There's provincial legislation in place to make sure they aren't damaged, they aren't harmed, they aren't stolen, they aren't sold, etc. So field work involves a lot of photography, it involves a lot of sketching, it involves a lot of observing the environment around where the specimens are, basically collecting as much information as possible in the field from the specimens actually in situ. We're not allowed to take them out unless they're potentially in danger. If they're in an area that's close to a coastline and they're actually working loose, we may be able to extract them to save them. But by and large, 99.9% of the specimens in the field stay in the field.
For example, if they're detached from a fossiliferous surface, like in a big slab or something that is vulnerable to tides or something, in that case.
Exactly. Rather than it getting washed out with the tides and being lost, we actually have an opportunity to save them. But those are all very strictly regulated by the government, so that they have to go into a central repository. In this case, it's at a museum in St. John's called the RUMS. They're the university's and the government's official repository for fossil material here in Newfoundland.
Right. Okay. And but yeah, we don't extract the fossils, but we basically produce replicas of the fossils that we can maybe discuss a little bit about.
Yeah, of course.
So this will be the third question. So, well, we're gonna now jump onto the second question. Once fossils are found, what are the next steps? How do we paleontologists prepare, analyze, and interpret these specimens?
Well, the fossils here in Newfoundland and at the Discovery Geopark, a lot of time is actually spent in the field photographing the specimens, both to get as good as possible photographs of the images themselves, but also to get images showing the relationships of multiple organisms, how close they are to each other, which organisms are actually associated or found on the same bedding planes, etc. So we try to collect as much information in the field as possible using photographic techniques. But some specimens, especially the ones that have a three-dimensional relief, will actually make replicas of them using a high-quality food-grade silicone that allows us to actually make a cast that replicates a mirror image of the specimens themselves. When we take those silicone casts back to the lab, we can actually make hard replicas using plasticine or using jesmonite, which is a special plastic material, or even something relatively simple and cheap like plaster of Paris. We can actually make exact duplicates of the original specimens that we make casts of in the field. Those allow us to actually look at science with these specimens in much greater detail.
Right. Okay. So it is very clear that casting is an essential technique for us, and it's been for quite a while. So why is it still used today? Why has casting survived throughout time, and how does it compare to other maybe newer digital methods?
Well, we still cast specimens today because by and large it's still really the best way to make exact replicas of specimens that are in the field. We can do better work in the field than we could 10 years, 20 years ago, certainly if you go back further in time, using digital techniques. But to actually make an exact duplicate of a specimen in the field, and that gives you the option of being able to manipulate that specimen. You can use it under different lighting conditions, you can use different photographic techniques, you can even get into other things which we'll cover in some detail after. Digital methods are becoming better and better. They're becoming more and more important as paleontological techniques develop, especially fieldwork. There are some very, very high resolution laser scanners and LiDAR scanners that can be used to both make very nice copies of specimens in the field, as well as large assortments of specimens, larger bedding plane spaces. So again, we can start getting an idea of individual specimens and the details that are preserved, as well as relationships, physical relationships at least, between different taxa, different specimens, how many are in a given area, how close they are, if they overlap, etc. So digital methods are good and they're getting better, but sometimes it's good to have a physical specimen in your hands because it changes the way you look at a specimen. Looking at an image on a computer screen, being able to rotate it in 3 dimensions gives you a really good perspective. But that's not quite the same as actually having it in your hand, being able to play with it under different lighting conditions, different types of light even. Natural sunlight compared to fluorescent light compared to unidirectional LEDs. They all give you a very different take on these specimens, and a lot of the time they provide you with information that you didn't even know was in there, which is one of the fabulous things about having real replicas in your hands.
Right, and these techniques are not mutually exclusive. They can be used together. Like you can have a cast and then also implement whatever digital methods you're working with, scanning or whatever, so that we can do everything. In fact, in our group we basically operate with LiDAR scanners and also casting, so it's just we use everything. Okay, so this is linked to question 4. Modern technology has changed how we study fossils. How do 3D scanning and digital reconstructions enhance our understanding of Ediacaran organisms?
It's interesting looking at the material from the Ediacaran here in Newfoundland, because most of the specimens, in fact, almost all of the material we're looking at impressions of specimens on a sediment surface. We're not actually looking at body remains. So when people are working on dinosaurs or on fossil shells or with plant material, etc., a lot of the time you're not looking at the original material, but you're looking at a 3-dimensional replica of the original specimen. Sometimes the upper and the lower surface, sometimes internal surfaces. With most of the material that we're studying, we're looking at the footprint on the ocean floor. So we're looking at one surface. Using 3-dimensional techniques, using 3D scanning, using digital reconstructions actually allows us to take these impressions and start creating the actual structures of the organisms that made them. So all of a sudden, instead of just having a 2-dimensional impression, we're actually starting to be able to recreate 3-dimensional organisms that are creating these footprints. And that involves a wonderful combination of creativity, ingenuity, and hard science. Some people look at these things and say, well, you know, it's just a pretty picture. It's fancy, it's not real. But if you can actually look at a dozen specimens in the field that show specimens oriented from different directions, from different angles, etc., if you can create a 3D model that can explain all the variation that you see, you've probably gone a long way towards recreating what the actual morphology of that organism was. If your model only explains beautifully preserved specimens, it doesn't involve unusually preserved specimens, then your model is lacking. And this is one of the beauties of it, is I know from personal experience with the work that we've done within the group, creating models that actually explain unusual specimens, the atypical ones, the damaged ones, the badly preserved ones.
Right.
If you can include those within your model, your science is much more likely to be real, or as close as real as it can get when we're looking at something where we don't have the actual bodies preserved. But the 3D techniques that we can do allow us to really recreate a much more realistic image and a much more manageable image. It's not just a mental picture. We've actually got something we can manipulate in 3 dimensions. And that gives us a lot more power with what we can actually do to recreating organisms that are extinct from a group that's completely extinct.
Right. It's interesting. Our group recently became more familiar with and more interested in 3D sculpting, kind of like what you said, like from a 2D imprint on the rock to a 3D geometry, basically bringing it back to life in a way. And that really proves that you also understand how the organism, like if you're able to produce the 3D geometry, you're conceptualizing the organism as a whole. You're showing that you're capable of reconstructing how this organism actually looked like. So it's not just a purely artistic process, it's just part of our intellectual interpretation of the fossils as well. And that comes with biological and physiological implications.
And that's actually a very good point, because myself, I know from my training, I actually went to university. I'm trained as a biologist first. I'm not a geologist. I'm looking at things from the perspective of the organisms rather than the rocks that they're in. And a lot of the time when people think of paleontology, they think we're measuring specimens, we're counting specimens. And we certainly do have to collect empirical measurements of what we have, and we have to count, and we have to measure.
Sure.
But I know for me personally, the real challenge here is to try to take something that's been dead for 500 million years, has no living relatives, and try to actually reconstruct how it lived, how it ate, how it reproduced, where it lived, how it moved, trying to recreate a living organism, or at least as close as possible. And a lot of these 3-dimensional reconstruction techniques that we're talking about with scans and with digital reconstructions, they allow us to get closer to that than we've ever been able to do before.
Right. So I guess this is related to the next question when we talk about 3-dimensional reconstructions and so on and so on. These fossil geometries, these 3D objects, can be used in modern techniques. For example, I guess this is a question, it's kind of like for us to debate. It's a technique that I've used a lot lately, which is computational fluid dynamics. Is this a tool that has traditionally been used in engineering, but that is now being applied to paleontology? So okay, how can you help us understand how computational fluid dynamics, or CFD, is used in order to study Ediacaran life and its interaction with the ancient environment?
It's a wonderful tool, and it's something, I have to confess, it's something I wish I knew better, but I don't have access to A, the time, or B, the computing power to do more of it right now. But what I've seen developing with this software over the last couple of years has been remarkable, because not only are we now able to start taking 2-dimensional impressions on a surface using practical paleontology, traditional tools combined with, as you say, digital tools, we're now able to start recreating 3-dimensional models of what these things look like. Taking those models, we can use things like computational fluid dynamics, and we can actually start seeing how these organisms would have interacted with the environment around them. So a lot of the organisms from the Discovery region are organisms that we believe lay flat on the seafloor. They didn't move, they didn't have muscles, most of them didn't have stalks or stems, they would have actually been flat.
Right.
And the traditional interpretation is, well, they were passive, they lay there, and as water passed over the top, they exchanged nutrients and oxygen and waste material with the water passing over them. Fine, that's a very simple concept, and it makes sense. But using computational fluid dynamics and flow dynamics, we can actually start to understand how different structures and different organisms will engage with different types of current. If they lay parallel to a current, if they're perpendicular to a current, if they're overlapping each other, if they're very close or far apart from each other, or even dealing rather than with groups of organisms, but with individuals, if we think about how high a 3-dimensional structure exists, if it's just a simple collection of branches that are close together, if they curl slightly, if they have slight spacing between them, we can actually start to play around with these slight variations and see how that will change how they engage with the environment around them. If water passes over slowly, if it passes over quickly, how will that affect how the organism can engage with it? And it's been fantastic to see how using these different models, using computational fluid dynamics in computers, has shown these organisms are incredibly capable of changing the water flow dynamics as fluid passes over them. And that allows us to think more about, okay, if this organism can do this when you've got a single specimen sitting there, what if we have a field with thousands of them? How is that going to change the fluid as it passes over the entire community? Or if we have collections of different organisms living in a community, how is that going to change how fluid dynamics work? Because if you have an organism that sits straight up and it's 30 centimeters high, how is that going to change things that are immediately in the sediment around it? So looking at CFD really is giving us a way of seeing these organisms almost as if they were living organisms on the seafloor. And again, to be able to do that with an organism that's extinct it's remarkable.
It's remarkable,
truly,
yeah.
Truly incredible.
Yeah, so I guess like the way I see it in a way, well, once you are able to reconstruct an ediacaran organism, for example, in 3D, you have two options. If you want to study how it interacts with the flow, like from a mechanic point of view or from a physical point of view, you have two options. Either somehow recreate this organism like physically and have an actual water tank and, you know, just have a water current ongoing and then just have like some sort of sensor and study the forces and so on, or do it computationally or virtually, which is what I've been doing lately. So it's essentially recreating, just picture like a virtual flow water tank, just a virtual tank with your organism in the middle of the tank, and once you simulate this digital flow, you are able to quantify, for example, the forces that are being exerted by the flow, like the drag force or the lift force, kind of like they are calculated for airplanes, you know, like airplanes are subjected to force, to forces like drag and lift, and these are things that we can quantify using CFD. And it's also, for example, we can also plot individual streamlines within the flow, which is basically as if you dropped a particle in the flow and you could track that particle along the flow, kind of as if that particle had a sensor and you could track it, as if you could track the motion of that particle. That is also something we can do, modeling either the streamlines of the flow or just the behavior of actual single particles, which is something that is being done now. And as you said, we can have an idea of how they modify the conditions in the what we call the benthic boundary layer, basically the conditions of the flow where they lift. For example, if you have an organism just erect up into the water column, it's subjected to drag, lift, and so on, and it reduces the velocity of the flow downstream. So it modifies the conditions that all the fossils, all the organisms could potentially be exposed to, right? So this is something that it could be argued that is kind of like one of the earliest examples of ecosystem engineers in a way.
Exactly.
So next question is, mathematical modeling and simulations are playing a growing role in reconstructing past ecosystems. How do these approaches help us test hypotheses about ediacaran life?
It's been fascinating to watch this change, because there are different schools of thought about how we reconstruct ancient organisms and the environments that they lived in. And using modern techniques is critical, but it's also important that we use traditional techniques to make sure we lay the groundwork properly. If you take a simulated population, for example, of organisms living on the seafloor, digitally you can start playing with things like, as we said before, whether or not they're upright in the water column, whether or not they're flat, whether or not they're parallel to the seafloor with space in between. You can start modifying variations in position, in location, etc., to see how the environment has actually changed around them, how the organisms will impact each other. So mathematical modeling and these simulations actually allow us to learn a lot about how both the fossils we see, the story they tell us, as well as implications of what variations in those organisms. But one of the most important things with it is the basic work, the skeleton, if you will, you know, the bare bones of it is, if you don't do your taxonomy right, if you don't do the morphology of the organisms right to begin with, the data that you get in the end is not very realistic, because essentially you're putting in incorrect data to begin with. Your final model is not going to be realistic. It's not going to be a real model. So it's a fantastic example of how modern technology is allowing us to do really incredible, interesting things, studying fossils in a way we've never done before. But it also relies on doing really, really good taxonomic work, really good observational work, really good hands-on visual interpretation of the data that's available. So you do good paleontology, you can start doing good work on the environments, the associations that you get. And it's allowing us to do things like, as we say, to see how organisms impact each other on the ocean floor. If you've got dense populations, if you've got widely spread populations, etc., using these mathematical models, we can actually see how they will impact each other in terms of flow dynamics, like we said before. So we can see if one organism changes the flow dynamics over a full population because it sits higher in the water column, will that affect oxygen availability? Will that affect food availability? Will that affect how toxins that are produced by the organisms as waste actually get flushed away? We've got a lot more power to be able to do that kind of thing than we had before. So using these wonderful new techniques is giving us so many options to be able to study these. And one of the good things with them is, and this also refers back to what you were just talking about with doing the computational flow work. If you have a practical model, you can do so much, but changing that, resetting it, reestablishing new parameters in it can take time, can take effort, can take money. If you do it on a computer, once you lay down your established framework, you can make modifications to that, A, more quickly, and B, with a lot less fuss and time and trouble. And presumably with a lot less cost, because you don't actually need to run these models physically. You don't need the equipment to do them, etc. So being able to do these things on computers are allowing us to do probably a lot more work and study variations in different models to see how they compare and how the organisms would probably actually be able to cope within these different models. So it's a chance, it's a chance to really reconstruct ancient ecosystems in a way that we can see what would work and what wouldn't work.
Right.
Which is something I don't think people even thought of being able to do 20 years ago.
No, for sure. No. Yeah, one of my favorite applications of mathematical modeling or statistical analysis in terms of idiacan organisms would be the deeper understanding of community ecology or community composition, right? Whether you have like the spatial distribution of fossils as well, whether you have aggregated or segregated fossils, whether that points towards competition or speciation, or and also to discriminate between populations that are driven by these classic theories in ecology, where you have like niche speciation, different ecological requirements, different fitness, or whether the populations are driven by random reproductive events, like more stochastic demographic processes, which is actually quite interesting in my opinion, and is quite new, and that's an application that I'm a fan of. Okay, so that was the sixth question. We're now on to the last two questions. Rod, you're almost there. Hold on tight. So the seventh question is, Discovery Geopark provides an incredible window into early complex life. What are some of the biggest unanswered questions that modern paleontology is trying to solve here?
Well, we've touched on a lot of those just in discussing these fossils here.
I feel like we've covered mostly everything.
What's the nature of the science? We get very excited. We don't just talk about one thing, do we? We always talk about the big picture. Studying the material at Discovery is incredible because it gives us an opportunity to study the oldest known complex animals anywhere on the planet, at least at the moment. We will eventually discover things that are older, there's no question about that. But these are the oldest known complex body fossils that we can find anywhere on the Earth. They are very well preserved, and they are preserved in high numbers. So that actually gives us, as we were discussing, the option of studying individual organisms, working out their morphology, how they develop. It also gives us a chance to study how they engage with the environment using these digital tools. And it also gives us a chance to see how they're engaging with each other in populations, both within species and multiple species living together. And one of the interesting things with these organisms, and one of the most frustrating things with them at the same time, is that they represent a group of organisms that are extinct today. So we can't actually look at modern analogs. We can look at modern parallels perhaps, but that's not quite the same. But the other thing with them is they actually represent an ocean floor community that we don't find anywhere on Earth today, because we have bioturbators, we have organisms that live in the sediment and dig through the sediment. We have organisms that move along on the seafloor in pretty much every ocean floor community on the world today, and no matter how chemical rich, no matter how hot, no matter how cold. So the population that we're looking at Discovery, it's a population that we can't see anymore. We can't actually look at modern parallels, modern analogs. So we're really trying to figure out how the first populations of complex organisms actually started to live, how they started to engage with each other, how they reproduced, how they ate, etc. So we're really looking at how modern complex animals really started to engage on pretty much every possible level with the environment around them and with each other, and figuring out how the first animals did what they did. That's a remarkable, big question. I mean, and once we work that one out, if we ever work that one out, because there's so many different sets of organisms, of course, and so many variations, we are literally looking at the beginning of life as we know it today. And that's one of the biggest questions in paleontology. That's fantastic stuff. Yeah.
I don't know what is the biggest unanswered question, but after having studied Ediacaran for like roughly 5 years in my case, I have this perspective on the Ediacaran that we have ideas on what these organisms were phylogenetically or taxonomically. We have some ideas or hypotheses on how they fed and how they reproduced or how they interacted with each other, but we don't have any definite answers. I think, like there are theories, but not definite answers. I don't know if there will ever be definite answers, but I think that is the big enigma, like trying to build towards a definite answer, or just trying to expand on the theories that we currently have that are by no means perfect. If you think of like the big Ediacaran theories, there's always a but. There's always there's always this could be, but what about this case? This could be, but what about this case? So I think that we're in the process of solving those big questions.
That's actually a very good point as well, because one of the things that a lot of people don't think about is people tend to think of science as it being about absolutes, about things being right, things being wrong, and disproving a theory and moving on to the next theory. But that's really not the way science normally works. It's normally a process of gradual building, and it's a process of refinement.
For sure.
Sometimes a theory is thrown out because it's wrong. That happens. But most of the time, and for example, as you say, you as a student worked on this material over the last 5 years. Before you, professionals worked on it, students worked on it, people were working on this material for practically a century, and we're still expanding, we're still improving. We've had theories that have been brought up that have been thrown along the wayside because as we've learned more, we've realized they were incorrect.
Or even brought back.
As we're moving further, we're getting better and better at refining. A lot of these questions we can answer in vague terms. We're getting better at working out some of the details. But that one of the problems is sometimes people are reluctant to let go.
Yeah.
People come up with concepts, people come up with theories, especially things that they work on themselves. And sometimes people don't want to let go. It's like, well, I put a lot of time into that. And no, I think that my idea was right. Yeah. That's not the way science works. If somebody comes up with a newer idea based on new data, go with it. You have to move with the flow. And that's really important. Pardon the really, really bad pun. That wasn't intentional.
Yeah. It could also happen that some theories are deemed incorrect for maybe 15 years and they are eventually brought back under a different light as well. So that can happen.
Absolutely. When aspidillas were first described in, was it 1883, I think it was? 1863? Most of the community around the world said, oh, they're not biological. They're water escape structures in deep sea sediments. Yeah. And it was more than 60 years later when people identified these structures associated with animals, when everybody went, oh, oh dear, he was right.
Yeah, and that happened not only with aspidilla, with other fossils from Africa as well. It wasn't until I think 1958 that they were like, the scientific community was like, oh yeah, these guys are onto something. Okay, so we're just going to finish with the last question, Rod. So looking forward into the future, how do you see paleontology evolving with even more advanced tools and techniques? What new discoveries could be on the horizon?
New discoveries on the horizon. Well, there's always new discoveries on the horizon.
Yeah.
And again, to go back to what I said before, I do like to repeat myself. It's important because there will constantly be new techniques as computing power gets better and more accessible, as software is improved, as people's approaches to using these software increases and is more widespread and improves. Like any tool, it's only as good as the person using it. And as more people use them, as approaches improve, we're going to be able to reconstruct and answer questions better. But it always involves going back to the basics of doing good fundamental paleontology to start. Once you get your descriptions done, once you get your taxonomy done, we can start actually playing with the data that provides us. And I don't, as I said before, I don't think there's any question that at some point in the future we will find earlier Ediacaran communities. Where in the world, I don't know. I'd like to be here in Newfoundland because obviously, you know, it's fun to keep things right here. But it doesn't matter scientifically. Once we establish earlier communities and find older material, it will start to answer some of these other questions. But also important, if we find material that's slightly younger, that also opens new avenues of thought, because when we start to combine ancient communities and start introducing more modern organisms into them, we start getting into that transition from the Rangimorphs of the Ediacaran into the Cnidarians that came afterwards, or things that came even later in the Precambrian, like once you start getting into the Australian material, the Dickinsonian and things like that. So important discoveries will always be based, at least in some part, on practical paleontology, being in the field, finding new sites, reinterpreting material that people have looked at before. But digital techniques, I think, are going to really, really become more and more important over time as people get more access to the software, as new software is developed, as skills improve, and people just learn to see things from a different light. That's always the core of what we talk about when we talk about improvements in science, changes in science, is what's happening up here.
Yeah.
And as people learn to look at this stuff with new eyes, come up with new ideas, there's no question we're going to come up with wonderful answers for all these questions. Some very simple things, but answering simple questions a lot of the time lays the groundwork for more advanced ideas. And that's one of the things I think that people need to keep in mind, is it's a combination of really basic stuff and then sort of taking it in any direction you can think of and asking questions, try to answer them.
Right. I think the only thing that I would like to add is, when we talk about technologies and softwares and stuff, I think the risk for modern scientists and paleontologists in particular is that they become, when we always talk about specialization, they become too specialized to the point that they become a tool themselves. You know, that you become some sort of software yourself, and you lose the perspective of your discipline as a whole. You lose that broader sense of knowledge that maybe more traditional paleontologists had. So my advice is for scientists and paleontologists to step out of their comfort zone and don't become a tool. Study different disciplines, different areas, and try to diversify your knowledge so you don't become, I don't know, a software. That would be kind of sad. So that would be my last message to the audience to finish with this podcast initiative. I hope that our listeners liked it. And well, Rod, thank you for participating in our last episode. I hope you had fun. And
thank you for the opportunity.
No problem.
I always love to talk fossils.
Our pleasure. And well, take care, Rod. See you soon. And bye-bye, everybody. Bye-bye.